So the next speaker is Juan Pablo JARAMILLO-CORREA from the Autonomous National University of Mexico, and he will be talking about genomic prediction for the management and conservation of small, secluded natural tree populations.
Thank you, Barbara. Okay. Thank you. So hello, everyone. Thank you for still being here. For those of you that do not know me, I am mostly working on phylogeography and detection of candidate genes. So this is me walking out my comfort zone and presenting the work performed by Sebastián, one of my PhD students, in collaboration with Andres, who did his PhD here in Sweden. And I'm going to show some results collected by Gustavo, another PhD student of mine, and Alicia, my partner in crime, working on the Mexican government. So let's see how it goes. Right, climate change. We have seen lots of projections from Europe. Sally just showed us some projections from Canada, and this is what's going on, or supposed to happen in the next decades in the central parts of Mexico, in the conifer forests that we have in the mountains there. So a drastic reduction of area and a moving uphill of these forests to the top of the mountains, to places where probably they don't have anywhere else to go. Cuauhtémoc science, a name that might be familiar to some of you, have predicted a shift, and we must shift some of these trees between 200 and 400 meters upward within the next 10 years in order to make adapted forests to the conditions that we are going to have in 50 or more years. And well, this is actually Cuauhtémoc planting some trees above the tree line in one of the mountains of central Mexico. This is an aerial view of the top of that mountain. And the main conclusion he has gathered from this pilot study is, well, that assisted migration is nothing that is straightforward. It depends on where do you take the trees from, the elevation, and how do you plant those trees. Below, nursing trees that protect them, give them shadows and stuff, or just by themselves, alone on their own. So mortality is far greater in these conditions than in these conditions. So assisted migration can be complicated. This is just a pic I took from the French network of Canada News last week of a collaborator of Sally's in UBC, who just put the thing quite straight. Reforestation is important, but you have to do it the right way. Plant the right species and which genotypes, the right genotypes, in order to have adapted forests to the future. And this is why we are here, if I am having misunderstanding all your talks. So, can assisted migration programs be guided through the selection of pre-adapted individuals? And can we use genome prediction models in naturals, would say Antoine, to predict which trees are going to adapt to those future conditions? That's the main hypothesis we try to assess in a pilot small test in one of the mountains near Mexico City. So this is the core of genome prediction. Most of you know that genome selection has been used in forestry mostly to reduce breeding cycles for mostly commercial purposes. Can we use these for conservation purposes? And the main idea lies here in the founding population. Founding population has to be small in order to create longer linkage disequilibrium between the genes we are studying and the traits we want to predict. Can we extend that to natural populations that are small and secluded, and we have longer linkage disequilibrium? So a little bit on the opposite way that we do traditional GWAS for detecting genome environment and genome phenotype associations. So let's see. We are working on furs in Mexico. Furs is Abies is a mostly temperate and boreal genus that sometimes decides to take a chance in the tropics. It's warmer there, making beautiful mountain forests that harbor some iconic species like the monarch butterfly in Mexico. They grow all along the mountains above 2,500 meters up to 4,000 meters in mountains which behave mostly as sky islands systems, mostly shaped by volcanisms and secluded after the last glacial maximum, now interglacial. So, this is just an example, again, from central Mexico, works by Gustavo, which shows that, well, isolation is quite strong, so strong that even some taxonomist colleagues have suggested to give different names to different genetic clusters based on morphology and ecology and other stuff, and strong genetic structure within things that taxonomists call with the same name. So, secluded populations checked. Demographic history, also done by Gustavo, indicates that there has been at least 2 strong bottlenecks which have drastically reduced effective population size in these trees, and the last of which has been lasted at least since the end of the last glacial maximum, so which have given selection to act and create some linkage between the selective genes and random genes in the genome. So, small effective population size checked. So, we selected one small place to conduct our pilot study. So, this is a natural park that goes around the Nevado de Toluca, which is a massive stratovolcano not far from Mexico City. And some of these lands are community owned, which is something inherited from the Mexican Revolution. Now the community owns and explodes the forest if there is ruins in them, when you have to pay the community in order to visit the ruins. If there is forest on them, well, they use the forest to produce wood. And one of these ejidos, well, they have been divided in different plots. They collect 10% of the volume in wood every 10 years. So they cut 10% of the volume, let the forest rest for 10 years, and then they come back 10 years later and collect another 10% of the volume. Problem is, I don't know how things go here in Europe, but firs are very lazy in Mexico. They do not regenerate fast. So some of these plots have been to be replanted, reforested with introduced seedlings. We use the oldest of these plots, which was cutted in 2012 and reforested 1 year later. And we measured, well, how the naturally regenerated individuals from that plot and the introduced individuals from that plot were doing. So we gave them some 5 or 6 years to natural selection to act on seedlings and saplings that were planted. This is Sebastian measuring things up, just to give you an idea of how tall are the saplings now. And he measured different morphological and physiological traits, just to have an idea of how individuals were performing. And, well, surprise, or no surprise actually, the reforested individuals were phenotypically different from the naturally regenerated individuals here in blue. Naturally regenerated individuals were taller and were physiologically more healthy than the introduced ones. However, genetically they were very similar and very distinct from neighboring populations, which says, well, oh, maybe there is some local adaptations taking place here. We performed this analysis with some SNPs derived from the Dirac experiments. Maybe we got over conservative here. We will have to lower a little bit our filters in order to have a little bit more genetic variation. Well, so this is the part I'm completely out of my comfort zone. I'm just reading. This is Sebastian's work. So he did an animal model with a marker-based relativeness matrix to predict phenotype from the genotype of the same SNPs I just showed you before. And he used those models to predict breeding values, which was compared with the phenotypic values in order to determine model ability, predictive ability, and predictive accuracy. I don't want you to read all this. I know in the back reading this is really, really difficult, but just look at the colors. The red ones are those who are below 0.8. All the rest are above 0.8, which means that either this thing is working or we have done something really terribly wrong. Heritabilities are in the range usually reported for conifers, 0.23 for water potential and 0.45 for stem diameter. So relatively similar heritabilities. Now, how these values compare with those obtained by people working on genome predictions in other forestries? Well, pretty well, actually. Especially when we compare to the only two studies I could find which have been performing natural populations. The one in white spruce, which is a collection of half-sieves from different mothers planted somewhere near Quebec City, and a flock of great teeth here in Europe, a large flock, trying to get prediction for breeding time. Very, very low predictive accuracy when compared with our own accuracies in Mexican furs. I am not going to fool you, I am not going to fool myself. Population size for both training the model and building the models are very small. Why? Because regeneration is so rare. I am not going to enter into the details here, but I would like you to concentrate in the two kind of green lines, if you can see them from behind. So this one here in the top, which are the models we constructed using the natural regenerated individuals from our plot to predict the phenotypes of the planted individuals in the same plot. And the other kind of yellow-green ones, which included some other naturally regenerated individuals from neighboring plots, a few kilometers away from our plot, to train the model. All of them, well, not here, in these four most important traits, were less performing, or performing less well than the ones constructed from the very local populations. This is another example of something that Andrew just showed yesterday. Adaptation and microenvironmental variation can affect quite a lot of population structure. This is an example of a story of the time when we were poorer than we are now. We used some AFLPs on populations sampled around a volcano in Mexico City. My student found a very strong population structure. After he performed some GWAS, he detected 9 AFLPs markers correlated with soil variation, and once he removed those markers, he lost all population structure, meaning that local adaptation could be driving most of the population structure we were finding around here. Please see that these blue lines are ancient glacial moraines, and this red spot here are lava flows from a neighboring volcano, so it is not the same thing for a tree to be here near the lava flows, or here in the top of a moraine, or here isolated and surrounded by pine forest. So we decided to take this to an even smaller scale within our plot. We took samples around 65 naturally regenerated individuals and 65 reforested individuals, samples of soils. We measured some edaphic characters, some of which here shown with stars were significantly different between groups. And we only retained the 4 that were not collinear with each other. And we used them to predict compound phenotypes derived from a redundant analysis. So we used edaphic characteristic and genetic eigenvalues derived from a Pinaceae. And when we use only the genetic eigenvalues, we could only explain less than 10% of the variation, of the phenotypic variation. When we included edaphic variation, we could explain up to 21%, almost 22% of the phenotypic variation. So including microenvironmental variation, can help us increase our phenotypic prediction. Let me just show you this. This is an interpolation of ammonia concentration in the soil. So trees over here are growing in far more ammonia concentration in the soil than here. And the genotypes of these trees, which are different, are interacting or producing different phenotypes, even if they are growing in soils with identical ammonium concentrations. So it is that bad. It is that bad. At the end of the day, well, we remain a little bit like Cuauhtémoc here, scratching our heads, because, well, the success and thriving of transferred individual is far more complex than just putting trees around. Let's plant 1 billion trees, say it's some governmental authorities. But we need to take account on ecological aspects, such as the ones Cuauhtémoc used, soil conditions, such as the one we measured, and maybe other conditions, such as mycorrhiza or climate effects or whatever. The open question is, can we take all these factors into account? Quick question, I don't know if we can. In Mexico, surely we cannot. Here in Europe, that's up to you to answer. So just to wrap up before I get red carded, yes, we think that genomic prediction models can be extended into natural populations only if these natural populations are small and secluded, and if we use local germplasm to build those models. However, microenvironmental and other traits should be taken into account in order to have more stronger models. And just a reminder for all of us, and including myself, climate change might be only the tip of this terra-deforming process we are doing to the planet. There are other things going on, such as pollution, which is killing massive forest fires near Mexico City. Let's keep that in mind too. It's not only climate change. This work wasn't possible with the help of all these people and the funding of all these agencies. Thank you very much for your attention.
Thank you, Juan Pablo. The floor is open for questions. Leo?
Yes, my question is, so now you are able to make predictions of several traits. Now it comes the question of, well, to say that it's an open question, how to combine those traits, how to use weights on those traits, which traits it's important in order to make the right selection. Have you have any thoughts about that?
I think that's more a question for you than for me, actually. I don't know. I was talking to a physiologist the other day who was visiting Mexico, and his very personal opinion, which I tend to share a little bit, is it the tree dead or not? I think at the end of the day, if we want to do assisted migration and we want assisted migration to work, the most important thing for begin with is that the trees are able to survive and to survive well. So drought tolerance, pollution tolerance, grow relatively fast but not fast enough to get killed by cold or whatever will happen in winter at high elevations. Is a very complicated question to answer. The quick answer is I don't know.
I have a question. I'm curious to know whether you have in Mexico a conservation strategy, in situ conservation strategy, and how do you do conservation in areas where you have also local communities highly affecting the sites? This is quite an interesting challenge.
That's another difficult question to answer if we want to be politically correct. Yes, we do have conservation strategies. However, the political instances dealing with conservations are kind of fighting to each other and getting into competence from each other. Because social needs in Mexico often collide with conservation needs. So it is difficult to conserve the forests when there are indigenous peoples who need wood in order to cook their food. And that is what happens in many places, even not far from Mexico City. So yeah, there are conservation strategies. Most of them are local, and most of the more effective ones are those who are performed including local communities. But Mexico is so diverse that it changes quite a lot from one region to the next and from one forest region to the next, and so on and so forth. So there is not an easy answer to that one either, but yes, there are some conservation strategies.
Thank you for the talk. Sean Hoban from the Morton Arboretum. Can you talk a little more about the pollution pressures that you ended your talk on? Are they changing the quality of the air or the soil? And is there anything we can learn from local adaptation to some of these pollution pressures?
Yeah, well, I went too fast at the end. I was a little bit stressed because of the yellow card I got booked, so, you know, like football players have to be careful. So that's another place, a different place from the one we are monitoring for soil variation and so on. So this is a place just next to Mexico City. So what happens is that we produce nitrogen oxides and stuff like that, that during the dry season in spring derives in tropospheric ozone, which accumulates in the southwestern part of the cities where the fir forests are. And firs are very susceptible to ozone and there is massive dying. You go into some places and you feel that you're getting into Mordor or something like that. It's quite depressive. There is some regeneration going on. Some of those trees show signs of tolerance, and we are concentrating in those. That's so far what we have been doing.